There's absolutely tons of stuff that is relevant to what you do. So it's about making the connections to what's going on right now and your course. And the connections are many. There's such a huge amount of information here, but as I said, it's supposed to be a revision. These are supposed to be stuff you've all done before. And so what I wanted to do, I will just share, with you some of my reading and how that relates to the A-level spec. And so that you'll see what you study on A-level is a sort of starting point for understanding what goes on. And I think the other thing that inspired me was the words of the Donald Trump, just so ignorant about science, and it would pain me if any of you were that ignorant. At least I hope you won't be as ignorant if you talk to people about this virus in terms of scientifically ignorant, but you'll see what I mean about that. So on your spec, what you have to learn about is the AIDS virus, okay? And so there's some similar things here. So that's, the virus is called HIV and the disease is called AIDS, okay? So in terms of understanding this, obviously this bit on the right you don't need to know, but so you can talk knowledgeably. What we're facing at the moment is SARS acute respiratory syndrome coronavirus 2, SARS-CoV-2. SARS-CoV-1 was in 2002. It caused 8,000 deaths, mainly in Asia. Okay, and so it's coronavirus disease 2019 as opposed to 2002. So it's COVID-19. No, there haven't been 18 other versions of COVID. Okay, for us to practice on. There's been one previous version that's similar to this one. So on your AIDS virus work, what are the features? Well, you've got these glycoprotein spikes, okay? And these allow it to attach to a binding site on a helper T cell, which is a white blood cell, as I'm sure you know. Other key features, of viral envelope, that's cell membrane basically. In here we have a protein capsid, and in here we have RNA. Obviously some viruses are DNA. Both AIDS and corona are RNA viruses. Okay, and in the middle of this AIDS, you've got the reverse transcriptase, which is a very unique feature about AIDS. And although AIDS was once deadly, one of the good things that's come out of the research is the reverse transcriptase, because we use that enzyme all the time in genetically modified organisms, as you'll learn in year 13. So if we compare that to coronavirus, here's a diagram. So very similar, we've got spike proteins there. We've got different proteins, carrier proteins. They would be there in the AIDS virus as well. And we've got these RNA sort of loops in the middle. There's no reverse transcriptase there, folks. So the spike glycoprotein on COVID will have a different shape binding site to the one on the AIDS virus. Okay, and that means it will bind to alveolar cells and some kidney, intestinal, and liver cells, whereas, as I mentioned before, AIDS will only bind to T helper cells. Okay, so AIDS is a disease of the immune system. Well, intestinal, to be honest, the coronavirus is not going to get through to the intestines. It's not going to get past the stomach. So the big, it could get to the kidney, but it's going to get to the respiratory system first. And so that's where we're seeing the problem, is it binding to alveolar cells in the body. So, and yeah, that's a more three-dimensional picture of that you may have seen of COVID, and they will be mapping out, as you'll see, for very good reason, they'll be mapping out the shape of these spike proteins, folks. We've been told to wash our hands. So what's that about? So the soap, the hydrophobic end of the soap here is going to dissolve into the cell membrane of the virus here. The hydrophilic end of the soap, it's usually a fat with sodium or something on the end, making this side hydrophilic, is going to bind with water. And that's going to smash the cell membrane and completely destroy the virus there. So it's, you know, it's easily killable. Well, can you kill a virus? It's easy to damage the virus. The virus is not alive necessarily. Some debate about that. Just to compare with bacteria, antibiotics normally destroy bacteria by damaging the cell wall. So antibiotics are not going to work here because there is no cell wall. And the other big difference, of course, is the virus is going to embed in humans. So if we look at the AIDS virus, it survives in water and other liquids. Okay, if it dries out, it becomes damaged. So the transmission of the AIDS virus has to be liquid to liquid, and the usual forms are semen, vaginal liquids, blood. Those are the usual ways of transmitting. So viral load seems to be relevant because we don't have any cases of people getting AIDS from kissing, although there is, you know, in theory, there's a route there to get it, because that's, if you kiss someone, that is liquid to liquid. But it appears that the amount of HIV in saliva is so low that we don't believe it's ever caused, we don't believe AIDS has been caused that way. And so the AIDS virus outside the body doesn't do well because it quickly dries out, and at that point it's not functional. So SARS-CoV-2 is different to that. It will last on surfaces for 1 to 3 days. It's undamaged in air for 3 to 4 hours. We seem to be able to catch it through the air. We seem to be able to catch it by touching surfaces and then touching our face. Okay, now there's been a lot of conjecture about, will this die out in the summer? You know, in general, some viruses denature if it's a bit warmer. You know, those of you, I don't know if in chemistry you've done Maxwell-Boltzmann curve yet, you'll know that temperature is just an average. You can have one particular virus in sunlight is likely to kill the virus. One part of the room can be an excessive temperature. That could kill the virus. So in general, we think that a lot of these viruses in the summer, they don't last 1 to 3 days on surfaces, so they're less transmittable. However, we don't, the thing is, I'm going to talk about COVID. There's a heck of a lot we don't know about that. And so if you look at MERS, Middle Eastern Respiratory Syndrome, that does not denature in really quite high temperatures. So, and I think the hopes of the summer killing off the virus might be receding. It seems to be fairly resilient at temperatures. Okay, so let's remind ourselves how AIDS works. We've got these protein spikes on AIDS, and they're going to bind with a receptor in the cell membrane. And if and only if they successfully bind with the protein on the cell membrane, they will enter into, remember this is the T cell, okay? So the virus will enter into the T helper cell if there's a match. And that match is called, it's incredibly similar to enzymes, folks. But you cannot call it an active site, but it basically is an active site. All the things apply, but you're going to call this a binding site, okay? So it's not lock and key, it's induced fit, but there's going to be a binding there of this protein spike with this receptor on the cell membrane, okay? And so that's the AIDS virus. So the RNA strand there will use reverse transcriptase to turn the RNA into DNA, and then the DNA inserts itself into the nucleus, okay? And, well, you can't really have a clever virus, but this is quite, this doesn't have a brain, but this is quite clever because it lies dormant there in the white blood cell. And the reason that's clever is because if you're not someone who's having an infection, you might not have that many white blood cells. And this virus only is able to enter T white blood cells. So if it just immediately, what it's going to do eventually is produce tons and tons of, it's going to take over the whole white blood cell mechanism and use it to make tons of viruses. That's the short version of all of this. Okay, but meanwhile, it will lie there dormant. And at that point, you're HIV positive, but you do not have AIDS. Okay, you're a carrier, if you like, for it. If you get a cold, there seems to be some kind of trigger to cause this to start making loads of viruses. And the reason that's quite a smart move is if you have a cold, you'll obviously make lots of T white blood cells. And so that gives the virus a chance to make, you know, thousands and thousands of viruses, because there's thousands and thousands of white blood cells at that point, as opposed to there only being a few. Okay, so that is quite weirdly clever, but it does mean the ultimate fate is that you, as an AIDS sufferer, you used to die of the common cold, because your white blood cells would be taken over with making viruses instead of mounting an immune response. So that is all vital stuff. Find it very interesting to compare with the coronavirus. I get this is beyond the syllabus, so you really don't need notes on this. So this is showing you the spike and the binding site of what's called an ACE2 receptor. And as we've said, that is in the alveolar cells, okay? And so one of the reasons we think this is more infectious than SARS-1, or if we're trying to determine how infectious a disease is, obviously we're looking at R0 values and all of that. But in terms of the mechanism for how something is more infectious than another, it's quite important to think about that. And so if it easily binds or binds more strongly to this receptor, we're going to say it's more infectious and there's less virus required to start an infection than if it's quite hard to bind to that. Okay, so the shape of this protein spike is what's going to determine how infectious this disease is at the molecular level, okay, and not much else. The other factor we're seeing with COVID is, you know, different people are having different responses. And so there's a lot we're not clear on here, but potentially people might have different numbers of ACE2 receptors in their body. If they have more, they might be more susceptible. Some people on heart medicines, low blood pressure medicines, end up with more of these. So that's one connection with what we call the comorbidities, which means that people who already have other conditions are more likely to suffer more severely with this illness. Okay, so what happens after that? As again, you don't need to know this on your spec, but it's pretty interesting. So we form a vacuole. So we've got a continuous cell membrane there, just as we do with AIDS. Sorry, I should have said that is a sort of endocytosis going on there, that it gets sucked into the cell. And then we can see the RNA coming out of the virus, and this time there'll be no reverse transcriptase, so it won't form DNA at all. It'll head straight to the ribosome there. Okay, so it'll take over the body's ribosomes, and as we've said, common ancestors, guess what? Viral RNA works with human ribosomes bound to. Okay, so viral proteins come out there, that side. Again, human RNA polymerase is what's used to clone the viral RNA, and then that cell in the alveoli is used to make thousands and thousands of viruses, which are then released by exocytosis. Okay, and so it's similar to AIDS, but AIDS, as I say, can lie dormant. AIDS, the RNA will embed in the nucleus. This won't. This will get straight to work So this is what inspired me to do this lesson. Okay, question 1. This is a very brilliant enemy, you know, it's a brilliant enemy. They develop drugs like the antibiotics, you see. Antibiotics used to solve every problem. And now when the biggest problem the world has, the germ has gotten so, I can barely read it. The germ has gotten so brilliant that the antibiotic can't keep up with it. That is our president of the free world. And as I say, most antibiotics, how do they work? They work on disrupting cell walls. Okay, and if you remove the cell wall, then water potential just does become an issue that can burst the cell or shrink the cell. The purpose of the cell wall is to stop water damaging the bacteria. This is nothing new in that sense. We've always had viruses, and they always are a little bit tricky for. They've never worked for antibiotics, and it mainly is that they get into the cell, don't they? And so you wouldn't want. You don't want an antibiotic that can destroy human cells. I mean, this would be mayhem. You can't use such a chemical. That's insane, because how would it kill only the infected human cells? That's the point. You cannot have any meds. Obviously, you know, acid would kill the virus. Acid would kill human cells, but you can't use acid indiscriminately. You just kill yourself.